Okay, so this is the lecture for our DNA extraction, and what we're going to need to do is go over the DNA lab itself. At the top of your composition book, I want you to title it The Stuff of Life, How is DNA Extracted? And I also want you to write down this learning target. I can explain the double-stranded complementary nature of DNA as related to its function in the cell. Okay, so I'll leave that up there for a minute. Basically, we're going to be extracting DNA from strawberries, and I'm sure you've heard of shows, that try to solve murder mysteries and this and that, and they use evidence at the scene to help convict murderers, rapists, whatever, of a crime. And one of the things that they look for is DNA evidence. So DNA tests are actually really important, not only for forensic scientists, which are people that do that, but also for biologists and doctors and detectives. So if you could just imagine yourself working in a lab where someone has brought a sample of strawberry from a crime scene, and you needed to test it. You needed to test the DNA of the strawberry. Once you would find the DNA in the strawberry, you would look for genes in the DNA, which are segments of DNA that code for something. You would use those genes to help identify the type of strawberry it is. Okay, but in order for us to actually identify the genes, we have to extract the DNA first, and that's what we're going to be doing in our lab. Okay, so the materials, I want you to write down the materials in your composition book. You're going to need a 50 milliliter beaker, one Ziploc bag, a coffee filter, 95% strength ethanol, which is like rubbing alcohol, and it's going to be really cold. You're going to get a whole strawberry, and you're also going to get a lysis buffer. And there's one more thing that's not on here. I'm going to type it in. I wonder if it'll let me type it in. One more thing that we need to add in here is we'll put a centrifuge tube and a beaker. Okay, so we need both of those things. And then the lysis buffer, like I was about to mention, is actually a combination of things. It's a combination of water, Dawn detergent, salt, and we mix it up, and that helps to break down the cell walls of the strawberry cells, because there's plant cells and animal cells, and plant cells have cell walls and they have a cell membrane, so you have to like break through those things in order to gain access to the nucleus, which is where the DNA is housed. So that helps to break that down and actually break down the nucleus so that the DNA inside the nucleus is released. Okay, so once you have those materials listed in your composition book, I then want you to write a prediction. I want you to predict what you think it's actually going to look like in the end. Okay, so go ahead and title it this part prediction, and then what it's going to look like. Okay, so far you should have a title at the top of your composition book right here. You should have the learning target written. You should also have the materials written, and then you should also have a prediction. Okay, so what we're doing is we're going through a lab, and so you're kind of writing like a little lab report. It's not a formal lab report, it's just a little lab report. And then your procedures are very important. This is probably the most important part of today's lecture, because you need to follow these exactly. If you don't, then it will not extract. The DNA will not extract. So the first thing you want to do, and you need to write these procedures down, and you can reword them so they're not so long. So I'm going to just verbally read through them step by step in a shorter manner, so that you can, get it done pretty quick. So the first thing you're going to do is get a strawberry, put it in a bag, and pulverize it. That could all be one step if you want. So get a strawberry, put it in a Ziploc bag, and pulverize it. Pulverizing it meaning that you're going to have a pencil or a marker, and you're going to smash that strawberry inside the Ziploc bag. Then you're going to go to the lysis buffer station, And at the lysis buffer station, we have graduated cylinders. I don't know if you've used those before, but they're basically a little cylinder that has little marks on the side that designate how many milliliters are in there once you pour things. So you're going to need 10 milliliters of that. You're going to add 10 milliliters of lysis buffer to your Ziploc bag, and you're going to zip it back up once that lysis buffer is in there, and you're going to take your marker or pencil and you're going to roll it over the smashed strawberry and lysis buffer combination for 2 minutes. Then you're going to take the materials in that Ziploc bag and you're going to pour it over a coffee filter so that the coffee filter traps the strawberry flesh, and the liquid part of the strawberry will flow through that coffee filter and then go into, so you want to make sure you have this over something, into the 150 milliliter beaker. You're going to let that sit for 10 minutes. After 10 minutes is up, you're going to take the filter, you're going to throw it away. Then you're going to pour the strawberry juice, if you will, and lysis buffer solution into a centrifuge tube, which is provided on your tray. And then you're going to go over to the ethanol station, and this is where we have ethanol sitting on ice. You're going to very carefully pour 30 milliliters of ethanol into that beaker. Now you don't want to just like dump it into the beaker, you actually want to tilt the beaker, and then you want to allow that cold ethanol to run along the inside of that beaker. So you don't want the ethanol to just directly hit the strawberry solution. You want it to run along the inside of the beaker as it's tilted and slowly pour that in so that you're literally like pouring it in so slow that a layer of really cold alcohol sits on top of the strawberry solution. Kind of like oil and water, how oil sits on top of water. That's what it's going to kind of look like. Then you're going to cap your centrifuge tube. If we don't have centrifuge tubes, we'll just use test tubes. And then you're going to let that sit in the beaker so that it's standing upright, slightly at an angle, for the next few minutes. What you're going to see, what you should see, is a precipitate occurring, which is something solid coming out of something liquid. And it'll be like white stringy things that will start to precipitate out of the strawberry solution and into the really cold alcohol. Then you're going to use either a glass stirring rod or a popsicle stick to get the DNA, and that white stuff that I just talked about, the precipitate, is actually the DNA. You're going to wrap it around the popsicle stick or glass stirring rod, and then you want to call me over to show me that you have successfully extracted the DNA. What I would like you to do once you have that is compare your DNA with the DNA of other groups. Discuss the similarities and differences. And then what you want to do is return all lab equipment. So the way that you got your lunch tray that has all the stuff on it is the way you want to return it to the back of the room. So you need a clean coffee filter, you need obviously the lab tray, a clean beaker, a clean test tube, a clean popsicle stick or glass stirring rod, and a clean centrifuge tube if I didn't say that, okay? So we also need to add to, I'm just realizing this now, we need to add popsicle stick, or you can also have a glass stirring rod. I'm attempting the popsicle sticks this year because the DNA would slip right off the glass stirring rod last year, so it won't, maybe the popsicle stick will hold on to that DNA a little bit longer. Then what I would like you to do once you have those procedures written is answer all of these questions. There are 7 questions, okay? And so I would like you to answer those questions in your composition book. Again, you can listen to this lecture over and over again to make sure that you get these questions written. I would like you to, I would like you to write these questions out and answer them. I'm not opposed to you printing out this sheet and then cutting it and tape and gluing it into your composition book. That's another possibility to make it easier. Okay? All right, so that is our pre-lab lecture.